Hello everybody, my name is Rolando Sanchez.
My name is Marissa Messina. And I'm Danica Morrison. Our research project is on the indirect predator effects on feeding behavior of kelp forest invertebrates. We would like to acknowledge our mentor, Christy York, our PI, Robert Miller, as well as the UCSB Marine Laboratory, the JKC Foundation, Santa Barbara Coastal Ecological Research Team, and CNSI. Very few people know what fuels one of the most biodiverse and productive ecosystems on Earth. Kelp forests provide food and shelter for thousands of species, from plankton to carp to leopard sharks. In addition to this, kelp is also an active member in our carbon cycle. Kelp forests are responsible for 75% of the ocean's carbon fixation. Kelp is not only essential to marine life, it is also important to us. Kelp is an economical powerhouse. China alone reports producing 250,000 metric tons of kelp annually. There's an ingredient in kelp that can be used as a thickening agent in products such as toothpaste, shampoo, soap, salad dressing, and even ice cream. Because kelp supports marine life, it also supports seafood industry. Lobster, tilapia, and bass are just a few of the economically important organisms that thrive within the kelp forests. The interaction that we will be focusing on is that between the rockfish, Idotea, and kelp. Idotea is a pinky-sized invertebrate that feeds on kelp. It has many predators, but in our research we'll be focusing on the brown rockfish, just to observe the predatory effects. We're interested in this system because our hopes are to collect data that can help us create predictive models to better understand the effects ecological disturbance might have. For instance, what if the predators of Idotea disappeared? Then the Idotea would be unchecked and have the ability to run rampant on the kelp population. Idotea are small, but they can eat more than half their body mass in kelp per day, which could pose a problem. So this is an example of a direct behavior effect. The population of Idotea directly affects the health and biomass of the kelp. What we're interested in is finding out if there's a non-consumptive indirect behavior effect. Say if we add a predator, such as the brown rockfish, could it have an intimidation factor on the Idotea, perhaps causing a change in its feeding behavior on the kelp? If this is the case, then the rockfish would have an indirect behavior effect, and that's what we're going to be looking into.
So we conducted the experiment in a tank that has sea salt water. And our rockfish serves as our independent variable, our kelp serves as our dependent variable, and our Idotea serves as our control variable. We put one blade of kelp with 3 Idoteas in the clear plastic container. The clear plastic container serves as a protective barrier so that we don't get direct behavior effects, that we're only looking at indirect effects. And each tank has a treatment and control. The left, on my right side, has the treatment with the presence of fish, and then on the left has the absence of fish. Essentially, these 4 boxes are all the same. These 2 have the 1 blade of kelp and 3 Idotea, and then these 2 boxes have just 1 blade of kelp and no Idotea. So that serves as our control. And for that reason being is because kelp degenerates naturally in water, so therefore we can compare our results. And initially, before starting the experiment, we weighed the kelp, and then after 24 hours of running the experiment, we visually see that the Idotea did graze on the kelp, and therefore we can see the difference in mass and compare the 2 treatments of the presence of fish or absence of fish statistically to see if there was an indirect behavior effect.
So in order to actually visualize our graphs, we use a box plot, and what it is we have these darker lines going through these boxes. The box is separated by this line, and you have 25% on top and 25% below. So what that is that's 50% of most of our data points are going to be in there. The line right here, the dashed line, also represents 25% above and then 25% below, and that's known as our extremes. In our x-axis, we have fish absent control, fish present, and fish present. In our y-axis, we have here is kelp consumed in milligrams a day. And what we have when we look at the first box is we have Idatea here, and they're consuming kelp every day. And interesting in our second box is that some of our data actually falls below the 0 line, which indicates that we believe that some of our kelp actually started disintegrating and then pulled in water, so it actually gained some weight, skewing some of our results in this one. Our third box plot here, if you notice that its median is very similar to the first one, and we'll get back to that because we haven't factored in Idatea body mass. And our last box plot is no Idatea present, and that's what we're expecting. What happens is if we take the amount of kelp consumed a day and then divide that by the actual body mass or biomass of Idatea, we notice in the absence of fish that they will actually consume about 80% of their biomass every single day. But if we actually reduce the fish, then they're only going to consume about 40% of their biomass per day, which is very significant because there's not a lot of experiments and research going on in marine ecology that has to do with indirect effect. So because our preliminary data does suggest that there is an indirect effect, that because the rockfish, just the presence of the rockfish alone will change the behavior of the Idatea, it may favor the health and biomass of kelp. And also, because our data does suggest that there is a correlation, in the future there's going to have to be more experiments done so that we gain more confidence in this indirect predator effect theory. And then also, because studying these food chain relationships, we can get a good understanding of what's going on and what actually supplies the oceans with nutrients and energy. Thank you.